Hello everyone and welcome to week 3. My name is Catherine and it's about 12:25 on June 3rd, and I'm going to be discussing about the microscopy of microbiology, the use and function of the microscope, along with 2 other items. I'm going to do this as quickly as possible, so I'm not going to do every single step. I'm just going to do the basic minimum because it will be time consuming for 3 exercises. Okay, so first I'm going to start out. I'm going to identify the components of the microscope. And first we have the eyepiece, the arm, and then we also have the turret. And then we have the objective lens, which is the big red one here. And then we have the condenser lens, which is the smaller one. Stage clip, to hold the slide. We have the stage itself. The diaphragm, which changes. And the illuminator, the light, off and on switch for the light to go off and on. So like so. And then I have the adjustments, the knob adjustments. And then we have the coarse focus knob. And I'm sorry, this is the fine. This is the coarse. Okay. And then we have the adjustment knob for moving this back and forth or moving it forward and backward. Okay, and then we have the base, and I believe I mentioned them all. Okay, now to get started, first I'm going to describe the field of view, and that is an area where it's being observed. So you're able to see just the image itself through the microscope. And the depth of field is the distance, range, focus at time, which is emphasizing the area of more of the background. So you're going to see everything. And the next thing, I'm going to observe pre-served specimens, the protozoans, the fungi and the bacteria. So here I already did the pre-served samples. So I have my bacteria spirillum, my bacteria baculus ammoba whole mount, bacteria coccus forum, yeast whole mount, and then the one that's preserved also is the penicillin with candida. And these, you can all put them on the microscope like so. So I'm going to just do a sample with the penicillin with candida. And I'm going to put it underneath. And this actually holds pretty nicely. And if you look through, it might help if I turn this on. And I'm going to try to put it on. Let's see. What do I do? Having a little bit of trouble with the, there we go. Yeah, see, when you do this, this is using the observed to make it more. And then if you want to make it further away, like I just did, and you can adjust it too, using the coarse focus or the fine. So I can make it clearer. That doesn't help. Okay, perfect. So you can see that that's the penicillin candida with candida. I'm going to try the bacteria spirillum. And I'm going to do it using the power is in the middle between the observed power and the condenser. And we have to use the coarse to adjust it. So you really have to adjust to see where it's visible. There we go. Perfect. Okay. And that's how you would do that for that one. And then now I'm going to prepare a wet mount, and I'm going to prepare for my cheek cells and my dental tartar. So I have my sterile cotton tipped applicator, and I have my mounts ready. So I did one with green, and I did another mount with blue. Okay. So the green one, I'm going to use that for my cheek. So I need to put it, I'm going to actually go inside. Okay, and I'm going to put it on the green one, which is this green one right here. I hope I got a good part of the cheek. Okay. And then I'm going to put a drop of the distilled water. And I'm going to use one of the little tiny squared slides to cover. So see, I did put a drop on here. It's hard to show you. And then, oops, it's moving around. And then I'm just going to stick that on. And then I'm going to view that one now. So I'm going to put that under the slide. And as long as you are able to view it using both of these is good. But to use your immersion one, this is like 100 power. You could switch it out so you get a really good view. And you're just going to put that one on. So I'm going to turn it like this. Okay. And you have to readjust this one as well. Wow, you get a really good view, like I said. So it's 100% power. And you can put a drop of the oil there too. So you put a drop of oil. So I'm going to take this out, put a drop, and then that's how you would do it. And that's just for the cheek. Now for the tartar, okay, and here I have the blue one still waiting here. I'm going to use this for the tartar. So hope I get something. And I'm going to put that on there. Okay. And then again, I'm going to put a drop of the distilled water. And then put one of these little plastic covers. The slide cover, the protector cover. Like so. Okay. And then I'm going to put this underneath too. And I'm actually using the 100 power. Oh wow, see? You get a good view. So this does really a good job. And you can adjust however the lighting, sometimes the lighting needs to be adjusted to make it better. Okay. And there you have it. And that's for this exercise. And following that, I will go on to the next part. And hopefully you were able to understand and got a quick view. Until next time, thank you.